Here are your meiosis video notes. So it's really important, I want to note before we get started, that as we go through these notes, when we get to each stage of meiosis, are going to need to either pause it and draw each stage, or you could pause it and maybe screenshot each stage, but you're going to need pictures of each stage in your notes. So let's talk about the basics first. So meiosis is a cell division which produces germ cells, otherwise known as sperm and egg cells. And in this situation, one parent produces 4 daughter cells. Now this is different than mitosis, which only produces 2. So in meiosis, there are actually 2 divisions. There's meiosis 1 and meiosis 2. As a result, those daughter cells are going to have half the number of chromosomes as the original parent cell. So what is a gamete? Well, a human gamete is a sperm or an egg cell, and they have 23 chromosomes. But humans have 46. How is that even possible? Well, 23 chromosomes are in the egg and 23 chromosomes are in the sperm, which after fertilization results in one single cell with 46 chromosomes. So a cell with n number of chromosomes is considered haploid. So example, eggs or sperm each have 23 chromosomes, and a cell with 2n chromosomes is considered diploid. So for example, the parent cell has 46 chromosomes before division. Here's just a quick little comic for you. So it shows mom with 23, dad with 23, and then the child has both of those puzzle pieces to give him the 46. So there's other things chromosomes can tell us. Things such as a karyotype. So a karyotype is a picture of all the chromosomes of an organism. But what do you notice? Well, look at the sizes and look at the shapes of each. So as you start at 1 versus 22, you notice the shape changing quite a bit. So we start out pretty large here and pretty small here. You also see X and Y, which are the sex chromosomes, which we'll talk about more in a minute. So how are chromosomes paired? Like we mentioned, by size and shape is how they're going to be paired with each other. But you also notice gender. So males are XY and females are XX. There's also things called chromosome disorders that can be evident in karyotypes. So Down syndrome, for example, there's an extra chromosome at 21. Turner syndrome, it's just X. There's not a second sex chromosome. And Klinefelter syndrome has 3 sex chromosomes. So here's a look at some of those. So Down syndrome, as you can see, there's an extra chromosome at number 21, so there's 3 there. Turner syndrome only has 1 sex chromosome, so there should be a Y, but there's not. And Klinefelter syndrome has 2 X chromosomes and that Y chromosome, so there's 3 sex chromosomes total. Now that we've talked about that, let's get into meiosis. So during meiosis, DNA is going to replicate once, but the nucleus divides twice. This is something called reductional division. The parent cell is diploid, and the resulting cells are haploid. Why do we even do meiosis then? Well, the reason for this is because it provides variation. That's really, really important. That's the whole reason why we do sexual reproduction. You get genes from 2 parents, which gives you a variety. The more variety there is in a species, the more able they are to survive certain things that may target only one gene or one trait. So we'll get into that a lot more when we get into the evolution unit, but the variety is really important. Something called crossing over can also occur, which we'll discuss in this video. And the gametes randomly combine. So there's lots of different factors as to why sexual reproduction is a variety and it's better. Asexual reproduction, on the other hand, is when all organisms come from a single parent. So it's basically a clone. That new individual is genetically identical to the parent. Homologous chromosomes is a term that you also need to be familiar with, and these are referring to pairs of chromosomes with approximately the same size and same banding on the chromosomes. So that size and shape that we talked about, they're pairing up into homologous pairs. Now that we've talked about that, let's get into the divisions. So like we mentioned, there's 2 divisions. There's meiosis 1 and meiosis 2. Meiosis 1 is known as reductional division, and meiosis 2 is known as equational division. So here we go. Meiosis 1 is that first division of meiosis. So interphase is the same as in mitosis. That the chromatin is going to condense into chromosomes, and the chromosomes are going to replicate in the S phase of interphase. Prophase 1, though, looks a little bit different. So those centrioles still are going to start forming the spindle fibers, which is the same as in mitosis, but the chromosomes find their homologous partner, and together they form something called a tetrad. So let's review. What's homologous? So homo, prefix meaning same. They're the same size, same shaped chromosomes that pair together. This is also where crossing over can occur. So crossing over is when chromosome segments or pieces of genetic information are exchanged between a pair of homologous chromosomes. As we just mentioned, homologous chromosomes equal a tetrad. So why do we even do this? Well, it adds more genetic diversity. So here's a look at what that looks like. So you have your two chromosomes, a paternal and a maternal, and you see crossing over. They're basically exchanging a limb on each other to where now they have that different variety. And then when those chromosomes are pulled apart later in meiosis, they're going to have a little bit of a different recipe, so to speak, than they would have originally, giving even more diversity to the offspring. Now that we've gone through prophase, let's look at metaphase. So metaphase 1 is when the spindle fibers attach, which is the same as in mitosis, but this time they're lining up double file. They are not in single file, they're in their pairs. Then we move on to anaphase 1, which is when those spindle fibers shorten, they pull them apart, but remember, they're pulling apart the pairs. So these are entire chromosomes moving to the opposite sides of the cell, not chromatids yet. In telophase 1, the spindles are going to break down and the chromosomes unravel, same as mitosis, but we have two diploid daughter cells and they have the same number of chromosomes as the parent. Then we move into something called interkinesis. So this is the step between meiosis 1 and meiosis 2, and it simply means between divisions. So there's no cell replication here though. It's not like normal interphase. It's just kind of a holding point before it moves on to part 2 of meiosis. So in the second division of meiosis, you're going to notice it looks a lot like mitosis, except there's two cells. So in prophase 2, this is the second phase of meiosis beginning. So that spindle reforms, we reform those chromosomes, and they're centered kind of in the middle of the cell here. In metaphase 2, they're going to line up single file in the middle. So this is starting to look a little bit like what you're familiar with. In anaphase 2, the spindle fibers shorten again, and this time they pull those chromosomes apart into chromatids. This is where we get the chromatid that you're used to when you think of cell division. In telophase 2, the chromatids are going to reach the poles, and they're fully separate at this point, but the cells are still not separate, remember. Then we go into cytokinesis, and in cytokinesis, there are 4 haploid cells. Very important that you remember this, because they have half the number of chromosomes as the parent cell, or N number of chromosomes, and the parent had 2N. So eggs and sperm are very important in this process. This is how they are created. It's also pollen or ovules in plants, so they are the reproductive cells. But there's a difference. In female meiosis, there is only one usable cell. So you saw in cytokinesis we just made 4, right? But in females, only one of those is actually an egg. The other ones are something called polar bodies, and they're not usable by the body. In male meiosis, all 4 of those cells are usable. It's also important to note that in female egg production, the egg is going to stay in interphase until ovulation, which is then when the cell goes through meiosis. And as you know, ovulation starts at puberty. So females are born with all the eggs they're ever going to have, but they don't go through meiosis until you reach that certain point in your life. And fertilization, as we know, is when an egg and a sperm combine. They make 46 chromosomes because they each bring 23 to the table, and that's going to form your zygote. 2 other things we need to discuss are male and female meiosis. So female meiosis is known as oogenesis. This is where we get that egg and those 3 polar bodies that are not able to be used. So as you can see here, we have meiosis 1, which is kind of occurring before birth, the after birth line. And then meiosis 2 is when that fertilization happens and we get that egg cell. So look through this, maybe pause here. This is a really good graphic of how this process happens. Spermatogenesis in males is a little bit different. So males, like we said, they get 4 that are all usable. So all of these sperm cells are usable. And those are going to be made, once again, through meiosis. But just important to note that females, only 1 is usable. Males, all 4 are usable. So there are also some problems that can happen in meiosis. So there's something called non-disjunction, which is failure to separate is what this word means. And it's when the chromosomes don't separate. So if you look at this picture here, you see this cell. This is after meiosis 1. We're left with 1 chromosome, but it doesn't split into those 2 chromatids like it should. They both stay in the same cell. So as a result, you get a cell that has an extra chromosome, and that's where things like Down syndrome happen, for example. It's also shown down here some genetic disorders such as Down syndrome, Turner syndrome, Klinefelter syndrome, all result in either having N+1 or N-1, and that could be the sperm or the egg cell that is fertilized, resulting in a child with one of these disorders. And that's all for your notes.